In the following presentation, the results of Figure 4 will be discussed. In this figure, the authors show that Hrd1p is not involved in the recognition of Hrad-L substrates. They also show that the 3A mutant that they discovered in Figure 3 is only deficient in the degradation of Hrad-M substrate HMG2p and does not affect the degradation of other Hrad-M substrates. In Panel A, an experiment similar to that used in Figure 3 was performed. Here, cells were transfected with variants of the Hrd1p protein, chased with cyclohexamide to stop further protein expression, and quantified to observe degradation over time. This time they measured the degradation of a luminal protein called CPY, which follows the Hrad-L pathway. In this case, a null strain was used as a positive control for no Hrad. In the gel, see the degradation that occurs with the wild type and the 3A mutant Hrd1p protein. This is indicated by the decrease in intensity in the lanes after 90 minutes. On the graph below, it can clearly be seen that the positive control was impaired for the Hrad degradation of the luminal protein, indicated in red. This was expected not due to any involvement of the Hrd1p in recognition of Hrad-L substrates, but rather because Hrd1p is needed in the Hrad-L pathway for ubiquitination of substrates. Thus, by deleting Hrd1p, no substrate can be targeted to the proteasome for degradation. The results show that the 3A Hrd1p mutant, indicated in the red circle below, resembled the wild-type degradation of the CPY luminal protein. So in panels B and C, they basically did the same experiment but using two different luminal proteins, and they saw the same results. So in conclusion, even though the 3A mutations to the Hrd1p protein significantly inhibited degradation of the Hrad-M substrate HMG2p, all the luminal proteins they tested displayed no difference in the degradation compared to the wild-type Hrd1p. The authors wanted to test multiple Hrad-M substrates to see if the 3A Hrd1p mutant was universally impaired for all Hrad-M substrates in Hrad degradation. As you can see, the positive control used was the Ubiquitin Ligase that was non-functional, the C399S Hrd1p mutant. However, an unexpected result was seen. The HA-PDR5 Hrad-M substrate showed the wild-type degradation. This was one of their most important findings in the whole paper and led them to believe that perhaps the Hrd1p protein is involved in the specific recognition of each Hrad-M substrate, and led them to move on in the paper and investigate this further in the following panels. In Panel 4E, the authors wanted to look at the function of the 3A mutant as a ubiquitin ligase using a growth assay. To understand this, you need to know that strains contained a mutant Sec61 protein called Sec61-2p. As you know, Sec61 is a crucial translocator for life. However, the 2p mutation makes the cell become sensitive to temperatures above 30 degrees Celsius, since at this temperature, Hrd1p mediates the degradation of the Sec61-2p protein. However, if the Hrd1p is non-functional, or unable to ubiquitinate the Sec61-2p protein, then the mutated Sec61-2p protein will not be sent to the proteasome for degradation, and the strain will be able to survive even at the higher temperatures. Observe the image above of the fluorescent cultures being serial diluted from left to right. The fifth row down, you can see that the non-functional C399S Hrd1p control was able to live at 37 degrees Celsius. In contrast, the 3A Hrd1p mutant was not able to live, resembling the wild-type phenotype. This was indication that the 3A Hrd1p mutant was able to ubiquitinate the Sec61-2p protein and thus is a functional ubiquitin ligase. So now that we know that the Hrd1p 3A mutant was indeed functional for ubiquitination, we can cross this off of our list as a possible explanation. Therefore, the 3A residues of the mutant Hrd1p could be involved in the recognition of HMG2p protein. Also in panel E, the authors confirmed the results of their growth assay by their normal cyclohexamide chase approach. As you can see, unlike their positive C399S mutant control, the 3A Hrd1p mutant resembled the wild-type degradation of the Sec61-2p membrane protein. So, what did we learn in Figure 4? Well, we saw that the 3A mutations to Hrd1p did not affect the degradation of luminal proteins in panels A, B, and C. In panel D, it was expected that the PDR5 membrane protein would show impairment in degradation similar to the HMG2p protein. However, the 3A mutation did not affect the degradation of PDR5. In panel E, the authors used two different techniques to measure the degradation of another membrane protein called Sec61-2p. Again, no change in degradation was seen from the wild type. By displaying that the 3A mutant was indeed a functional ubiquitin ligase, the authors gave supporting evidence that the 3A mutation was affecting the recognition of the HMG2p protein. In all, because of the surprising results in D and E, it was hypothesized that the 3A residues of Hrd1p were specifically involved in the recognition of HMG2p misfolds.